Good afternoon, all of
Good afternoon, sir.
you. Yeah, welcome to this session. AML formulation framework. So I took this peptides and buccal films, water scripts related to nutraceuticals. So this is a companion to the drug discovery desk. So same philosophy applied to the formulation also all the 12 stages. Here the peptide buccal film from target product profile all the way to commercial validation with a deployable digital twin. So doing the prediction. Here the contents will be here also 12 stages, stage execution to so reference and closing. So TPP, QTPP, CQS, and API pre-formulation, excipient cell screening, DOE, design of experiments, manufacturing, dissolution modeling, and permeability modeling, stability modeling, analytical AI, master ML pipeline, digital twin, and scale up and validation. Here, how to use this framework is every stage shares the same internal structure, purpose, inputs, and critical parameters, procedures, and data schema. The data schema is the procedure, data collection, pre-processing, EDA, and model building, next output. So, for example data and the ML model and the decision output. So the key idea front is the stage one quality attributes become the objective function for every downstream model. Here, here the master flow is 12 sequential stages with a closed active learning loop. The stages 6 through 11 stream batch data, so back into the master data set and the digital twin, so which retains the score candidates like rating. The 1 to 10 are in between the 1.5, 0.6 are nearby 1. Score candidates and proposes the next experiments about 30 to 50% fewer runs than the full DOE based upon the results. Here the stage execution. Here the 12 stages themselves each with its data sets, models and deliverables. Clear? So what are the features should be selected for each phase? We'll come to know. Here, the stage one, the contract between the formulation, the patient, and the regulator. Okay, so every model treats these targets as its objective. So weak profile makes the model optimize the wrong thing. The deliverable is signed submission grade document. So every document regulatory anchor for stage one, ICH, the quality documents Q8, Q9, and Q6. Okay. So the purpose, the inputs. Input, this is indication and pharmacology data. Target patient profile, age, swallowing, ability, and compliance issues. Competitor product analysis, Orange Book, and EMA reports. API IP status, so patents exclusivity, regulatory pathways. 505B1, 505B2, ANDA, NDA, OTC. So here the QTPP elements. In QTPP elements, the buccal peptide film for polystroporosis. Each row is a binding criteria. 500 micrograms, one daily, onset under 10 minutes. 24 months shelf life. That's what good means for everything downstream. Here, for example, if you see a different peptides, for example, like a BPC, GHKCU. So the BPC is for body protecting compound and the GHKC is for skin glowing. So these doses for the oral route, for example, like a buccal route. For buccal route, what time it will take to dissolve inside the buccal. So what are the enzyme degradation like in the saliva? So what are the factors it will affect to dissolve in the, or to lose, what amount it will lose to the dosage. So these things will be evaluated. And the CQA definitions like critical quality attributes, the variables for the model. Here, the CQA assay, content uniformity, dissolution, tensile strength, holding endurance, moisture content, mucoadhesion, permeability, total impurities, oxidation impurity, microbial load. So, when you generate the data to feed the ML model, so what kind of data sets, so the future should be there. For formulation, for this like analytical point of view, once you've done the formulation, when it comes to your analytical lab, so these parameters you should evaluate it. Based upon these results, again you go back to the formulation has to be optimized. Proper ingredients or excipients, how much quantity need to be added and which included or which excluded. Here our assay is 100%. So limit will be 95 to 105. HPLC or UPLC or LCMS method. This will be like critical. Risk will be critical. The patient input is sub a super therapeutic dose. Here, so the main criticality, the problem where it will come, the stability issue. When I prepared for 1 mg dose, okay, so today I prepared and I evaluated assay. It's like from 99% it came. Okay. Then after 1 week, if I put my product in the different conditions, for example, minus 10 degrees Celsius or 2 to 8 degrees Celsius or room temperature. So in three different environments, I stored my sample. Again, after 10 days, again I evaluate with my API, freshly prepared working standard with particular ppm. Okay, so when in my previous result, SA I got 99%. Once after evaluation of 10 days, if my minus 10 degrees, 2 to 8, if I getting 80%, and my room temperature stability, if I getting only 40%, then again I has to need look into the formulation because minus 10 degrees and 2 to 8 degrees, my formulation up to some 10 to 5% loss will be there. But in my room temperature, my loss will be 40 to 50%. Then again I go back to the formulation. So any stabilizers needs to add or any permeation enhancers need to be added. So these things should be evaluated. So if I design DOE, if I design my design of experiments, I hello, hello, yes ma'am.
YouTube link.
Prakash, sir, sorry
Yes
for
ma'am.
the interruption.
Yes.
I will mute everyone, sir.
Okay.
Kindly give your
నేను యూట్యూబ్ లో కనెక్ట్ చేయలేదు దీంట్లో ఓకే ఓకే యూట్యూబ్ లో చూసుకుంటా చేస్తాను.
Yes, sir. I have muted everyone. Sir, please take over the session.
Yeah. Sorry for the interruption. Okay.
We
So
are sorry for
essay,
the
okay, essay. When my essay were evaluated through HPLC or LCMS, okay. So LCMS different approach will be there and LCMS different approach will be there. So I have to see the column, the mobile phase conditions and my diluent. So these things should be evaluated. So in a lab, every time has to check it out every experiment to get the results. So before going to get the results, when we're using the AIML, I can predict exactly 80 to 90% what the column or mobile phase or what conditions if I use it, I will get it the optimum product. Then here 60 to 70% of time I can save. Remember, when you collect the data, then you start the experiments. After getting the results from the experiments, again you have to be designed in a proper way. You should get the good results. Not like every time you have to do it the experiment and again modify it and again do it. It's not like that. When AIML comes to the picture, this all the data should be tabulated in the form of this way. Okay, so content uniformity, dissolution, disintegration, tensile strength, the folding endurance, moisture content, MC, how much it is there? Either 8% is there or 10% is there water content, and muco adhesion, permeability. So permeability is, for example, if my peptide is less than 500 delta, then according to the rules, the rules of permeability, actually BCS class, which class my drug is? BCS1, BCS2, BCS3, BCS4. In BCS1, high solubility, high permeability. BCS2, again, high solubility, low permeability. And BCS3, low solubility, high permeability. And BCS4 is low permeability and low solubility. So these things will be, you have to be segregate your peptides like that. Excuse me. Sorry for the interruption. So this BCN1, BCS1, BCS2 or BCS3, BCS4. Okay, so if your peptide is and comes at BCS3, high solubility, low permeability, then what to do? What the changes you have to be done in your formulation. Thus you have to be evaluated. Okay, so total impurities and oxidation impurities and microbial loading. Total impurities. For example, if my HPLC analysis, if I my retention of time is getting around 4 minutes, if I my run the sample around 2 hours, then suppose at 1.5 hours, if I get some peak, maybe it will be reducing my assay. Then I have to look into that what is the impurity. Either it will be like from the solvent or from analytical method or from the synthesis. So these all things should be evaluated. Next. Risk estimation. So in critical quality attributes are the Y variables of every model. You should evaluate the risk assessment for assay. So polymers, okay, the assay uniform content and the dissolution, tensile, moisture, muco adhesion, permeability, impurities. So which factor is more risk or less risk? For assay, polymer is the low strength. Low risk is low. The polymer percent also low, which is high API load. If my API, if it is I'm getting impure API, then my assay won't pass. And dry tea, solvent and pH. pH also. So when pH, normally in human body, 37 degrees Celsius and pH will be neutral, around 7. So if my peptide is around 6.8 to 7.4 or 7.2, in between. Sometimes if my peptide pH is, if in preparation of a formulation, if I am able to get my pH around at 6, if I am getting 3, okay. So before going to the tablet form or strip form, we won't get exactly final finishing product because my pH, it alters the entire formulation. So when you are doing a formulation, you should definitely look into the pH. That's what SI will be, the pH will be risk is high. And dissolution. Polymer is high, polymer percentage is high, and plasticizers is medium, and API load is the medium, and dry T is the medium, and pH also medium. So these things you should be tabulated. Once if you are going to the experiment, in experiment, in formulation, so you know the formulation, what type of contents you are mixing, how much percentage you are adding. So these things you should be tabulated. And risk factors also you should be tabulated separately. Then only you will get a clear picture. So what factors you should consider. Okay. So here critical quality attributes the assay, the content uniformity, dissolution, permeability, impurities, each has a target, a limit, a method, a risk level, and a patient impact. These are measurable and gated. The risk matrix and desirability. So here the risk estimation the risk estimation matrix the risk estimation matrix tells us which inputs to study in DOE. The high risk ones become factors, and composite desirability collapses many quality attributes into one number because it's a geometric mean. A single bad attribute crashes the whole score. Clear? So you should very thoroughly focus on this attributes, which is low, which is high. Next, the key formulation. Here, for peptides, the stage prevents 80% of late failure. So we characterize physiochemical properties. Physiochemical properties predict dissolution, permeability, and dose. The stability, the set storage, I said the -10, 2 to 8 are room temperature, and container and formulation pH. The solid state form, set crystallinity or hygroscopic strategy. Whether it will be like hygroscopic in nature or crystalline in nature. So what are the inputs? Greater than 5 grams pure API, reference standards, force degradation. So H2O2, HCl, and NaOH. Calibrated DSC, TGA, SPRD, FTIR, HPLC, UPLC, LCMS. So these instruments should be used to evaluate the different parameters. Okay. So this test batch, how you will do it? Molecular descriptors in silico. So like a smiles. The compound actually what we are reading, the system won't read that way. So they have a different nomenclature will be there. It will be smiles. So you should load the smiles. Then you should read the formula from the Python code. Then only you should, or the structure, 2D or 3D structure, you should evaluate it. Then only you can check it out how it will be. The stability profile, partition coefficient, hygroscopicity, and solid state force degradation under different ICH rules. Okay, next. Molecular to in silico method. So the compute everything we can construct. Okay, the compute everything we can from structure, from the structure, molecular weight, RD kit descriptors, molecular weight, log P value, RD kit, log D value, pH 6.8. So Marvin sketch or chemoxon, TPSA, RD kit, HBD or HBA, hydrogen bonding donor, hydrogen bonding acceptors, RD kit, rotational bonds, pKa, and charge at pH 6.8. So these the tools should use. This is the descriptors and this is the formulation. So if it's the molecular weight, permeability, sealing, Lipinski rule, how it will apply to this log p value, mucosal partition, whether this my compound have an ability to partitioning this mucosa. Actual ionization at buccal pH. So what is the pH should attain at 6.8 or 7 or 6? Okay, TPSCA, permeability indicator, hydrogen bonding to polymers, conformational flexibility, pH formulation strategy, mucoadhesion compatibility. So these things, we should be the code snippet source RT kit. So this is the code. See, this is the code snippet. From RD kit, import chem. From rdkit.chem, import descriptors, Krimple, Lipinski, smiles. See, this is the smile, this is the compound. This is the compound name, hem for long peptides. Mol is chem moles for smiles. Then, if descriptors, if you write these things from for this molecule, these all the details you will get it. Molecular weight, log P, EPSA, HBD, HBA, rotational bonds, heavy atoms, and aromatic rings. See here in coding section and all, see here the double double quoted molecular weight and this what is the structure it is there. Yes, such you should write it. No single space also. Single space if you given, then the code will give you wrong way. It won't execute. So that is the difference between the IT people and the pharma people. Pharma people normally you will write it simply way, but when you execute the code, these all the things you should be uh, concentrate. See here the bracket. What is the bracket and what is the this is everything will be in order. You should not mismatch the order as such as to be execute in the coding. Next here the solubility partition hydroscopicity. The wet lab basics solubility across the pH range. So buffer's pH range, for example, 1.2, 2, 3, 4.5, 5.5, 6, 6.4, 6.8, 7.4, 8, 9. So these all different pHs and access API to 2 ml each. Different pHs. Sonicate 5 minutes, heat at 37 degrees and 100 rpm for 24 hours. Centrifuge 15,000 grams for 10 minutes. Filter 0.2 ml PVDF. Free check API absorption. SA by granulation. So, like this, you should generate the data. So, initially, in solubility only, you will get the exact result. The compound, if it is a pure or not, or APA, how it will be, how much content it is there, you will get it. For example, GHKCU is a GHKCU is a skin glowing peptide. If my API I won't get it pure, then if I check in solubility test, my compound will give you actual original compound will give you copper blue color, thick blue color. If my API is not good, it will give you light blue color or some different color. Then only I come to know my product is not good or not up to mark in presence of potency or assay. Before going to assay, these basic test will get a complete picture. Next partition, shake flask method, free saturated octonal buffer, pH 6.85 Becquerel. API in 1:1, 30 minutes, equilibration at 25 degrees. Assay both phases. Log D is log C octanol or aqueous. So this log D at application pH is an operationally relevant number, not log P value. Hygroscopicity. So non-hygroscopic, slightly hygroscopic, moderately hygroscopic, very hygroscopic. Okay. So these tests you should done in your lab and you should maintain one data set with different properties because when you are evaluating the results for a particular compound, this data set will be used helpful for AIML model building. Clear? Next. The solid state and data set. So thermal and X-ray analysis tells us crystalline versus amorphous. The peptides usually prefer amorphous to solubility, but it's less stable. And here the capture schema, every property in a structured CSV, every property in a CSV, structured CSV, okay, comma separated, that is Excel. One more Excel, that is different formation. ML ready from day one. So the peptides are amorphous, hygroscopic, and charged. Everything downstream follows from that. So you should tabulate the form, tabulated the data. API, molecular weight, log P, log D, TPSA, charge. These are the physicochemical properties. It won't change at all. These are actually basic constants. For a particular compound, it has a particular value. So, so this schema, this is called it as a schema data set. So you should evaluate this data set. Clear? Next. Here CR plus force degradation. So we can predict permeability from the structure with a model. Like random forest as a baseline, graph neural network, GNN. We have enough molecules. For forced degradation, deliberately stresses the molecules up to 5 to 20% breakdown, just enough to identify the pathways. See, this is the predict permeability and solubility from the structure. This is the code. From SKLN ensemble model, import random forest. This is the model, algorithm. Under this from SKLN model, selection import cross validation score, import pandas as PD. When if you are doing this pandas, this is the one of the Python libraries. When you want to do structured data interpretation, we should use pandas. If we are using numerical calculations, NP, okay, NP you can use it. These are the libraries. With the help of these libraries, the Python execute the code and will give you the results. Okay, so here different methods is the cross degradation, stress, condition, time, and target degradation. So you should evaluate the results and you tabulate this model. Algorithm mapping is like a boost. This is one of the model based on structured formal data. Random forest, robust, interpretable, and good baseline. So the basic things you should know when you are applying in models, whether ML model or deep learning model. The basic things you should know. For which model have the characteristics to get it the input. What are the inputs I will get it? Gaussian process gives uncertainty critical for actual learning. Graph neural network. When the molecules greater than or equal to 10,000. Okay. Next. Peptide degradation pathways. The main problem is for peptides is the main stability. The stability, if you can maintain or evaluate the stability, then it will be huge impact will be there on every aspect. My resolution, my assay, my permeability, everything will depend on this peptide stability. So the peptide degradation, each degradation pathway has a mass fit fingerprint. We track in LCMS plus 16 plus 16 for methionine oxidation plus 1 for deamidation. So like different ways it will be there. Crucially, each pathway maps to a defensive design level. Oxidation means add methionine and EDT. Hydrolysis means keep water out. So like this, you should evaluate the temp. Next. Excipient screening. So now we select the excipients by function. So like polymers, number of polymers will be there. For which molecule, which polymer is suitable. Some basic literature such as to do. And again, any existed data or existed articles are there has to go through it. Then according to our formulation or our patent, we should select the excipient. The film format plasticizer, and permeation enhancer, mucoadhesive, and a peptide stabilizer. Each row gives an example, typical ranges, and the property it controls. It is like a film format HPMC E3, E5, E15. Fullam, PVA, or maltodextrin, sodium alginate, or cargrinan pectin. So typical range will be 30 to 60% weight by weight. The selection driver is tensile, dissolution, transparency. If the plasticizer, PEG 400 and glycerol, PEG, sorbitol, triazine, triethyl citrate, 5 to 25%. Like 5 to 25% mean, for example, if my product is, for example, 0.5 mg or 1 mg. For 1 mg, how much I has to add it? For example, my batch size is 20,000, 20,000 strips or 20,000 tablets. For each strip or each tablet, 0.5 mg. Then 20,000, how much it will come? Then accordingly, you should calculate the percentage and should be added. So these all things should be considered, and this the selection driver. These parameters you are focusing on particular flavor or particular sweetener. These parameters should be considered, and how much quantity be added in the formulation formula you should know. Next, so polymer library. In polymer library, we pre-catalog candidates polymers with great level detail. The point on this slide matters. So record the grade, not just HPMC. The reproducibility breaks you and drop the grade. For HPMC, E3, molecular weight K Daltons is 13. And VASC CP 2% is 3. Film quality is clear, slightly brittle. See, actually my product is, we are mainly focusing on the oral strips. That's what actually I designed in a, what are the required formulation will be there accordingly I designed. So tablet or any other formulation or other things will be there accordingly as to do it. And the dissolution will be fast. Within 3 minutes, within 5 minutes, my drug will be dissolved. So if 2 minutes, how much it will be dissolved inside? The brain blood barrier, it will be crossed directly, the in buccal root directly go into the circulation. So there is no loss in the my dose. Directly it will go inside. But due to the large barrier, I should consider, because of my peptide molecular weight, how much it will go inside. That has to be look into that. And the source, it's a Dow, and different sources are there here. So HPC, cytosine, pectin. So these all are formulated compounds which we are using in the formulation. Next. Black film screening. In black film screening, we cast blank films by standard protocol and run through a 12 test evaluation. So the visual, the thickness, the weight uniformity, the tensile strength, elongation at the break, folding endurance, surface pH, disintegration, dissolution, and thickness, drying time, and peelability. So these evaluation panel, these all things should be check it out. So my method will be black light. Clear or uniformly opaque, no bubbles. So when you are going to the formulation, each parameter should be evaluated. So solvent casting protocol, for example, I written like solution, 4% weight by weight polymer in water, ethanol or water, mix 500 RPM, 30 minutes at 25 degrees Celsius. Plasticizer, 15% weight by weight of polymer mass. Degas, vacuum 30 minutes or ultrasonic for 5 minutes. Cast will be 1 mm wet thickness on PET line. Dry 50 degrees, 30% RH at 30 minutes. Convection oven condition will be 25 degrees Celsius, 50% RH at 24 hours. Evaluate. See panel, right? Here, when you are doing my process, if my drying will be at 25 degrees, or 30 degrees, or 50 degrees, or 16 degrees, or 10 degrees, I should evaluate because my stability, once in different temperatures, I prepare my film and I inject my samples, then we check my assay. Depends upon the different conditions, my assay will be vary. So you should optimize the parameters, optimize the conditions, temperature conditions, humidity conditions, whether my product is stable at particular temperature. Clear? Next. Polymer screening data sheet. The see DOE, design of experiments. For example, here 10 trials, accepts and rejects. Whether the result should be accepted or the result should be rejected. So there is PS4. PS4 is accepted for this all trials. A pull-on HPMC 50-50 blend with PEG, good tensile, fast disintegration, and 82% release at 3 minutes. That becomes the skeleton for the next stages. So, like this, you should evaluate the schema and you should process the experiments. Okay, next. Plasticizer seal optimization. So on this skeleton, we swap plasticizer concentration. So here the decision rule is lowest moisture uptake while keeping folding and tensile in range, which lands at 15% PEG or triethyl citrate. For peptides, TEC is often preferred because non-hygroscopic and no oxidative breakdown. Okay, so here this PEC 400 is the accepted here. So what are the concentration, different concentrations, and the different tenses, elongation, folding, thickness, moisture uptake, and TZ degrees. If we consider these results, so this is the PEG will be 400 will be accepted. Next, API excipient compatibility. So the API excipient binary mixers under stress. This triphos is actually stabilizing. So glycerol and citric acid are incompatible. So this ML model, machine learning then does multi-output prediction to shortlist the formulation skeleton. And the models are saved and versioned. So this is the classification regression model, multi-output classification regression. So here I want to tell you, supervised learning and unsupervised learning. Morning I said you, supervised learning will be tabulated form. In supervised learning, actually two types will be there: classification and regression. Classification, in classification, like for example, different colors of flowers will be there: roses and red roses, yellow roses, uh, different flowers. So in classification section, each color of flowers or each characteristic of flowers, keep it aside. That is the classification. In regression, in regression actually, the concentration versus if you time, if you see in bioanalytical, the regression, the pattern, how it will be, either it will be aligned with the test, my reference and test, either compatibility is there or not. If my reference compound and my test compound, if my regression is one, both are compatible. That is like a generic to main compound. If it is not aligned with my main preference compound, that the regression in that regression, that is not good. So that is not compatible with the generic molecule, not compatible with my main compound. So in the classification models, in regression supervised learning models, it will give you this is the code. See, this is the polymer and polymer 2 and plasticizer, a ratio, plastizer and column transformer. So like this, you should give the code. First, you download this libraries and you should give the code and you should run it. Okay, then you will get the results. When you are using the huge compounds like 100 or 200 compounds, this will be very useful for you to analyze the compound. Clear? Next, design of experiments. Okay, in stage 4, in design of experiments, we replace one factor at a time with a structured design of experiment. Here, it captures interaction and curvature, gives a regulatory design space, and produces clean ML training data. ML training data, three phases like screening, optimization, and verification. Only high risk factors from stage 1 to be entered in this. Okay. The main effects of each factor: interaction effects, real source of surprise, and quadratic curvature for optimal location, high stage QA design space, and orthogonal points. So these three phases will give you a checkpoints. All the checkpoints. Next, a bracket bump. So a bracket bump screening. In screening, 8 factors. Okay, the 8 factors in 12 runs to find the vital few. See, like this, DOE experiments you should design like this. The factors and coded ranges, colon, low, high unit, total polymer, low, high unit, teraphores, permeation enhancers, casting wet thickness, drying temperature, and drying time. And this run PB matrix responses A, B, C, D, E, F, G, H. So different formulations, assay, how much it came, dissolution, tensile, and DIS. So different values you are getting. Clear? Understood? So these 8 factors into 12 runs. Identify vital few drivers. So what are the more appropriate results we will get it? Okay. Here, the rest get fixed at a safe levels. Next, box Beckman. Optimization. The 4 vital factors in 20 to 27 runs. Build a response surface for each attribute. Then we optimize composite desirability across all of them to find the optimum point under design space. The 3 confirmation batches verified. Clear? 4 vital factors, X1, X2, X3, X4, and in 27 runs. Low, mid, high. So these are what are the deliverables will be there. Phase A screening report, BBD design, and fitted RSM model, optimum point, and 3 confirmation batches, manufacturing and tested. Next. Stage 5, manufacturing. Here, process variability here destroys the model's predictive power downstream. So we lock it down. So 12-step solvent casting train with critical process parameters and acceptable ranges for each mixing up speed, pH, drying temperature, and oxygen headspace. So this solvent casting train steps, 12 steps. Solvent preparation, polymer dissolution, plasticizer, API solution, combine, deaeration, in-process check, casting, drying, and cooling, slitting, and socket packing. So these are solvent casting trains are critical process parameter, dissolution, RPM, and range, and why it matters, air, entertainment versus lumps. So for every step, you should note it down clearly. For my which factor affects for which and what up to what range. Then whatever the data you are entered in a tabulated format, in future it will be very helpful to you. A minimal or any unknown point of matter which will be there, it will be add on your model. and it will give you a good predicted model. Next. See, manufacturing data set. Manufacturing data set and a software sensor. Here, we capture every batch parameters and outcomes. Here, the batch, different batches, up to 10 batches. Scale, 100 grams, 100 grams, and 500 grams, 1 kg. RPM, and degree Celsius, and pH, and viscosity, and wet microns, dry zone, Z1, 2, or 3. And this one rust solvent, film, wet, assay, AV, and resolution. So these all parameters you should be tabulated. Because a gradient boosting soft sensor, predicts assay from process parameters. Then if it predicts, then you can go to the lab and you have to evaluate the experiment, then you have to check it out whether my prediction is correct or not. If it is predicting the correct exact value, then it will be applicable to all 90% of the remaining data. Because already you have to confirm with your lab data with the predicted value. Then you can confidently go for another remaining batches also, which eventually enables real-time release testing once a regulator validates it. Okay? So how scale and wet thickness shift the outcomes here. Next. Here, the dissolution method. So here in dissolution, the single best in vitro predictor of in vivo onset. So we pick an apparatus, a different model, set buccal relevant conditions. Buccal relevant conditions like a pH 6.5, 37 degrees Celsius, or PBS buffer with different pH. This is example I am saying. And confirm sick conditions so the kinetics are interpretable. So here USP1, USP2, USP4, different method for different when you use solid dose. Film spin is preferred for low dose, low volume. Baker or Petri dish discriminating simple bioavailability proxy. Texture analyzer in saliva is disintegration kinetics. This will be mechanism classification is thick films, thin films, or slabs. 0.5. Fickian diffusion, 0.5 less than N less than 10. One is 0.5 with anomalous diffusion or relaxation. One is case 2 erosion control. Greater than 1 is super case. So different conditions you should evaluate it and you should be save the data. Next. Dissolution kinetics models. Here, 6 standard kinetics models. Zero order, first order, Higuchi, Crossmeyer-Pepys, Weibel, Hexen-Crowell. So the different kinetic models, each fitting a different release mechanism. The code fits all of them and picks the best by R squared. Exponent in purpose model tells us the mechanism. So these are the different models with different equations. And this is the code. This is the code SIPnet has to use. Import numpy. Numpy is the calculations. See kinetics. Always kinetics will be calculated as speed of the reaction. So then in pandas, in Python, you should use numpy. So import numpy as np. Then automatically the system calculates each and every calculations in the dataset. For SkyPlay optimize, import config. So def0, temperature Kelvin, def first order, Higuchi, Pepper. So like this, if you build a code with these things, then it will be give you the result. See, this is the coding part. So this coding part, if you practice it in different V3 schools, then different online free open sources are there. You can try it in that. It will be easy. Okay, it will be easy. There is no much difficulty in that. Only the same pattern will be there. But for our chemistry, you should be implemented this one. See here how the code will be 0, 10, first 10.5. Higuchi Higuchi 30, peppers 30, 0.5. So this is the code section, and this will be pop it via R2 and accept exception as E out the name error. So this is the code. If it is any error will come, you should omit the error and has to give the code. So this is one part of the code. Like the different codes will be there. It will be check it out. Next. Here the profile fit ML F2 similarity. So F2 or F1 similarity or differentiation of that fits Kvsrscops Pepsi best. Pepsi's best. So one model it feels the best given the result with exponent around 0.58. So the diffusion plus relaxation. The F2 similarity factor here above 50 means the profiles matches. Okay. Is what we use for batch to batch and scale up comparison. ML predicts the full release curve for IVC. Okay. Here the fitting parameters, see the values. The result is given for this which parameter will be followed. And the F1001 resolution profile. S2 similarity factor, static ML, this static ML XG boost, and profile ML, LSTM. Okay, this is the one model actually. LSTM is the one model, different models. Next. Permeability method. Mucosal permeability is the rate limiting step for buccal peptides. We rank models by cost and transferability. So the Potkin buccal is the best practical XYO. Morning XYO. XYO predicator and run standardized France cell protocol to get flux and permeability. So France cell. Here the France cell protocol is France diffusion cell operator. Okay. In France diffusion cell apparatus, here the receptor donor cell will be there and the receptor cell will be there. In donor receptor and the water circulation will be there. It maintains the temperature, human body temperature. Once in donor compartment, so what is the relevant buffer? You have to put it like a PBS at pH 6.8, and in between donor and receptor compartments, in between you should fix your membrane. Either it will be Falcon membrane or synthetic membrane like a permeopad. Permeopad is a synthetic membrane. Then after synthetic membrane, your strip or your tablet has to keep it. Strip has to keep it on top. Then has to close it. Should not leak. Should not come from the system. Tightly close it. Then you start the experiment. Once start the experiment, once the permeability, the flux means how fast my drug can, how fast my drug release. Permeability, how freely my drug move, how freely my drug move. So, you should select the time points based upon your compound, your peptide. For example, like 5 minutes, 10 minutes, 15 minutes, 20, 30, 45, 60, 90, 120, 180, 240, 300, 360. Around 6 hours. 6 hours duration. At 5 minutes, okay, when you are revolving sonication, you are revolving that magnet, the water, the buffer will be revolved around your string. After 5 minutes, you should collect the PBS buffer at 0.51 mm. Okay, and you are separate it in your separate vial. Then how much you collected from the donor compartment, that much same buffer need to fill in the France diffusion cell. For example, at 30 minutes, 5 minutes, 1 ml you collected, 10 minutes, 1 ml you collected, 15 minutes, 1 ml you collected. At the same time, once you collected, at same time, same quantity, you should add it in the donor compartment cell. Again, the experiment will be continued. So once you collect the sample, For example, let us say 0.5 mg, 0.5 mg in 20 ml. So if you calculate, if you do calculations, if you do 400 ppm, you will get it as a stock solution. But your linearity 100%, if you selected around 10 ppm, then again your sample should be converted into a 10 ppm compatible to relevant to the standard 100% matching. So your sample. Then every time what you are collected has to dissolve, you has to dilute the sample at 10 ppm and you should inject. You should inject in HPLC or LCMS. So this is a permeability experiment. So PAMPA, PAMPA, liquid coated filter, TR146. So this is human buccal cell. This is the cancer cells. Normally, I said morning also, synthetic cells, permeable cells, in vitro, ex vivo, in vivo, and the cell lines will be primary cells and secondary cells will be there for CACO2. For CACO2 and this TR146 cells are there. These are the cancer cells, primary cells. So these also the transfer experiment has to do it. How my drug will be behaving in TR146 cells before going to the human clinical trials. Before going to human clinical trials, in vitro transdiffusion cell, in vivo in the sense like a TR146 cells. These all experiments has to do and collect the data, and based upon the model run ML, deep learning or ML models, if you run it, and you get the results from the based upon all the results compilation, you should go for the in vivo experiments. Clear? Next. So example permeability. The example of the permeability. Here, the screening permeation data set hybrid. The permeation enhancer sodium glycolate gives the best enhancement here. The model predicts permeability from molecular to formulation features, but we wrap it in a fixed lab. A mechanistic hybrid that extrapolates for more safely than a black box and satisfy the regulators. Here, the batch and enhancers. These are the different enhancers. So, sodium glycocolate, non-cytosine EDTA, sodium trichlorate, labrosol, SLS, sodium glycocholate. So, these are percentage, different percentage on film, the film thickness, and flux, and the pack. So lag time. So fast drug inside and pack the cumulative percentage of release of the drug at each time interval and enhancement ratio. So these are experimental data. Like this, you should generate the data set. So enhancement screening that exhibits for fixed class or hybrid model. You should collect the data in that way because this in future, if you are doing multiple products for the permeability enhancement or permeable experiments, this data will be you can use as a reference because somewhere it will be some physical properties or some different properties will be somewhere it will be matching or some variation you will get it. Based on the variation, these results if you feed the model and you can predict the unknown molecules, maybe the behavior of that one, how it will be. So the whole data that existed, what we have generated, are very crucial to the model building for the unknown molecules. Clear? Next. The stability methods. The stability sets the shelf life, storage, and the pack. The test across ICH climate zones. So the Indian and Brazil, the hottest regions, humid zones 4B. A long-term, intermediate and accelerated conditions. So the zone 1, 21 degrees, 45 degrees, 45% RH, humidity. Accelerated is 40 degrees, 75% RH. For use, EU and Canada. And subtropical is zone 2. That will be long term is 25 degrees Celsius, 60% RH. Intermediate 30 degrees, 65. Accelerated is 40 degrees, 75% RH. Japan and US. 3. 3 is hot. hot dry area. 30%, 30 degrees Celsius and 35%. Accelerated 40 degrees and 75%. Parts of MENA. Fourth, hot humidity is 30 degrees and 65% RH. And accelerated 40 degrees, 75% RH. So most of Asian countries. Fourth B, hot, very humid, 30 degrees and 75% RH. Accelerated is 40 degrees and 75%. India and Brazil. So the sampling points are the 0, 1, 2, 3, 6, 9, 12. These are the months, 1 month, 2 months, 3 months, 12 months, 18 months, 24 months, 36 months. Accelerate will be 0, 1 month, 2 months, 3 months, 6 months. So these different conditions, you should evaluate the stability of the product. Clear? What is the each time point will be? Appearance will be no change. SA 95 to 105. Total related RS, total related substance per ICH-Q3B. Specific impurities will be ID that should, or water content less than 5%. Disintegration less than 30 seconds, less than or equal to 30 seconds. Dissolution is Q plus 5 at 5 minutes. Tensile folding in spec. Microbial is per limit. And container integrity will be passed. So these are the stability parameters should be evaluated. Next, the stability data set. The stability data set. How you will see every stage you should maintain as a data set, as a Excel, or in different formats. If it is a different format, you should be converted into your useful format, which will be easy for the operations. One batch and this condition and T, SA, this TOT, IMP and IMP, this dimensions, water percent, disintegration, DS, disintegration and tensile. So these are the synthetic data generated from the for different experiments. So these are the data, like a foil socket, 4 conditions and multiple time points. Okay. So a real stability data set across the 4 conditions and many time points. Oxidation and deionization dominate the impurities. Confirm the stage to forced degradation, fingerprint, and the aggregation appears as the first condition for the past 12 months. Clear? Next. Algenius. Algenius ASJP hybrid ML. We predict shelf life with physics first. Algenius exploratory degradation rate to 25 degrees. The ASJP can predict a 2-year shelf life from 2 weeks of testing. Clear? Got it? 2 weeks of testing, you can predict 2 years of shelf life. So invaluable for trials. Then ML supplies only the residual on top of the physics. So this is the Algenius modeling. This is the code. This is the code will be generated and hybrid Algenius. So moisture modified Algenius. So this is the details. Has to enter. Next. Analytical. Page 9. Analytical AI. AI speeds of analytical method development. A chromatography DOE, DOE design of experiment finds the best conditions and a retention time model predicts where a new peak will land. So when an unknown peak appears, the model suggests what it is, collapsing days of investigation to minutes. Select, for example, if you give one molecule, in design of experiments, you should design in a Python, like a buffer range 2 to 7, organic this much percentage, 5 to 25 percentage, organic final 60 to 95 percentage for the retention and illusion for this gradient model. Gradient time 8 to 40 minutes. The column temperature 25 to 60, flow rate 0.2 to 0.5. Again, in column, different column bonding will be there, and the specifications will be there. 4.6, 2.1, 75 mm, 100 mm, 150 mm, 200 mm, or 2.7 microns, 3 microns, 5 microns. So these all parameters you should be tabulated in a tabular format. Clear? So when the pH range, pH buffer, like a 2.5, 3, 3.5, 4, 4.5, organic, acetonitrile or methanol, the composition 80-20, 60-40, 70-30, or 90-10, 95-5. So like this, you should give like code or in a Excel format, you should give in a different formats. Then you should run the total batch. Then which gives you the result, the most probable results it will give you. Then you can implement it in a lab, then you will get the results directly. Clear? Next. Impurity invention, impurity inventory or validation. We can even predict the impurity before running the anything in the system. A rule-based LLM engine, a rule-based plus LLM engine driven by the peptide sequence, validated against the force degradation peaks, and the method itself is validated against the ICH-Q2 checklist. For example, if you run in LCMS molecular weight prediction will be there. In molecular weight prediction, if you run full scan, once you will get the full scan, you can upload the full scan into AML model, and if you give the proper prompt, like what might be the degradation, what causes the degradation, what factors will be there, then automatically step by step it will give you what it went wrong, where it went wrong, what is the peak, either the peak is a API peak or a plasticizer peak or a polymer peak, which is come under this way or that way, based upon the molecular weight. Molecular weight, based upon the molecular weight, it will give you the impurity profile. If QTOF, if you use it, the amino acid sequence it will give you. The molecular weight will give you the LCMS ESA model. ESA, electrospray ionization. Okay, so here predict the impurity before running the total run. Next. So here the master data set. Master data set, all stage data sets. Okay, all stage data sets join in one wide table. Identifiers, molecular properties, physiochemical properties, formulation, different formulations, process CPC, CPPs, conditions, and CQS. And so examples of data set, you use data set you collect it in different entire different small experiment you did, and you collect the data and you finally mix it in a huge data set. Okay, so this is a master data set. So these all stages, this data set join in one wide table. The molecular descriptors, formulation attributes, process parameters, storage conditions, and the quality attributes that are the prediction target. This single table is what the master pipeline learns from. So this is the data. This is the data you generate, and you feed the ML, you feed the system. Then the ML model reads the data and the patterns, the different patterns which are, we won't see the patterns as humans. They can see the patterns, and from the patterns, they can predict the future results, what will be the, and that give you in a logical way, in a proper logical way, judgmental way, it will give you. Yeah. Next, future engineering. In future engineering, multi-target, how we engineer futures. So what we are there, these are all futures. These are all our futures. For example, if you say around 80 futures are there. In 80 futures, so if you apply your domain knowledge, whether some futures are really needed or that you can combine the futures. 2 to 3 futures, if you combine, it will give you one future. Then the remaining, whatever the unnecessary futures, we can eliminate it from the data set. Okay? So the future determines the future determines whether the model generalizes interaction comes cumulative process burden. So the keeping the time and storage separate. We choose a multi-target strategy. The common mistakes box is the practical wisdom. So here you don't futurize differently at train versus certain. Don't drop the slow high signal futures. So in future engineering, these parameters, step and the action, this multi-target modeling, one model per target, simple, easy, multi-output regression captures shared structure, one algo for all. So this way you can generate the model from the previous generated data. Clear? Next. Algorithms. Algorithms and stack. Here, in algorithms, the recommended stack picked by the data shape. The data shape is like how many rows are there, how many columns are there, what are the descriptive statistics are there, what are the descriptive statistics, mean, median, mode, what is my data set, whether my data set is in a bell shape, U curve it is there or not, whether my data completely is unbalanced is there, then how to balance it, then my data set will be like a trailing is there or fronting is there. Means in the sense skewness is there, positive skewness is there, negative skewness is there. Then how to correct it my data with my mean, median, mode, or my kurtosis and my skewness. So different statistical parameters will be considered, and you should clean the data and you should modify your data accordingly to fit for model. Clear? So here the always the linear and a tree base. Here the linear baseline, tree baseline, and boosting main, alternative boosting, tabular, deep learning, uncertainty, molecular only submodel, GLN. So these are the different models. We should the model runs with this algorithm. Data is there. For example, 2000 cells are there. Rows, columns, 2000 cells are there. From 2000 cells, from the existing data, these all algorithms run it, use the patterns, and which gives you the output. Whether what we're expecting, how much range it is there. It will give you. Okay. Next. Validation and sharp active learning. Here, three things. Here will be honest validation. Leave one formulation out is a realistic test. Sharp to explain predictions to regulators and audit tool. No decoration and an active learning loop that scores candidates by desirability plus uncertainty, reaching the optimum with 30 to 50% fewer experiments. So random k-fold, baseline reporting only. Striped is stratified by batch. Avoid leakage across replicates. Leave one formulation out. Closest to prospective use. Time split. Production realistic for the stability data. Nested CV. When tuning HP's honest performance. How you will tune, how you will do validation? For example, your data will consider the 100%. Then you can split the data 80-20. So 20%, 80% will be trained data and 20% your testing data. After running the data again, you switch the 80-20. 20 become 80 and 80 become 20. Again, you run the data. Then like that only, you should do the validation, cross validation. In cross validation, every aspect you will check it out. What is the input? What is the output? What the results it will be there. And then according to the what need to do with your data, whether further cleaning is required or further anything need to be added. Clear? Next. Digital twin. So the digital twin is a persistent virtual replica. You query in seconds. Okay. So it can pre-screen 10,000 formulas before any lab work. The layers stack mechanistic physics and ML ensemble a uncertainty and a design space check. An output with confidence bands and a sharp explanation. So In normally DOE experiments, like per day 10 or 20 you will do, or 30, 40 if you do have a good team. Then here 10,000 formulas at a stretch you can check in your with your AIML model. Input layer, user query, formula, process for storage condition. Mechanistic layer will be fixed permeability, Arrhenius stability, Cosmeyer resolution. ML layer, XGBoost, GP, GNL, ensemble per CQA. Uncertainty of design space, confidence interval, design space, membership check, output, predicted CQA, quality attributes, how it will be. Next, tech stack. In tech stack, an open source first stack and minimal fast API service, exposing a predict endpoint. You send a formulation, it returns each predicted quality attribute with a confidence interval, the overall desirability, and an accept or reject. So this is the code. Data store, data storage in a PostgreSQL, MySQL or PostgreSQL. So this is the data storage. How you are storing the Excel? Same way in structured query language, SQL. Structured query language, you can store the data. Okay, you can recollect the data and you can upload it in the system. Once suppose in different labs and different, this is the online data you are getting. Then it will be, you should always in a dynamic mode you should see that the data will comes in a way, then it will be processed again, the results will come again the data will come. So this is like a data warehouse. In Databricks, data warehouse will give you, in data warehouse you should different formats you collect the data. In different formats you should properly clean it and properly made it in a uniform dimensions, the data should be there stored with clean data. Then only you should process the data. Without cleaning the data, without different formats of data, you won't give the good results. For example, float, numerical value, numerical 1, 2, 3, float 1.2. So float will be different and numerical value will be different. So you should segregate it. Okay, that will be text format will be different. Numerical value, categorical data will be there, numerical data will be there. So you should properly see the data and the data type also you should mention it. Then only system will read clearly what kind of data it is there, whether the data should be converted into another format. Then only my model has to run. Next, scale up. Scaling from 100 grams to commercial. Variance compounds at every step. We match dimensionless groups for honest physical equivalence. Let the twin predict at new scale, bracket validate the experiments, extremes, and finish with 3 consecutive successful batches. So mixing geometry, risk lab. Impeller geometry, pilot will be larger impeller, commercial will be different vendor. Heat transfer, drying air pattern, surface to volume, operator interactions. So these are scale up to risk factors. You should evaluate the risk before going to the lab scale, commercial scale. Okay, next. Process validation, life cycle. So the FDA, here the FDA 3 validation stages map onto the framework. The modern twist is stage 3, continued verification. Continued verification becomes an ML problem. Stream every batch in, retain monthly and run statistical control on the prediction residuals. So you will get an early warning of drift, not a autopsy. So if before going to commercialization, if you run the model, then it will give you where any problematic is there, anything will be there which will be needed to optimize or modify, then it will tell you. Then accordingly you can modify and you can run in a commercial way. References. Here, ideal database structures. See, API CSV, the final master schema. Schema is like a data set. Different data sets will be combined and will give you. The final master schema, every CSV, comma separated. CSV is a comma separated. Query a table, every model version with a hash, every change audited. one key per table, so every measurement traces back to a single composition, process scale. No orphan data. So no spreadsheets floating in folders. Okay. So XCP and CSV like this every before going to the once we are step by step we have collected all the data. See XCP and CSV, formulations, batches, manufacturing, analytical, stability, dissolution, permeability, ML models, SA, DISO, stability, permeability, shop reports, audit reports. Everything will be an entire database. Okay. So in entire database, so model target algorithm, one key per table, and GXP ready audit. So in step by step, the ideal database structure should be maintained in that way. Okay. Next, software ecosystem. In software ecosystem, the full recommended stacks, chemoinformatics, peptide handling, classical ML, scikit-learn, class, and gradient boosting, deep learning, CNN for molecules, auto ML, uncertainty. So, and for uncertainty, these tools are very good to get the good results. And explainability, DOE, stats, visualization, MATLAB, Seaborn, Plotty, these are graphs you will get it. Pipeline tracking, serving, Bison optimization, and regulatory compliance. These are ecosystems, you will get the results. So these are the regulatory anchors you will get. So when you are doing the experiment, these regulatory guidelines you should follow. You must follow. Otherwise, it will entirely the process won't accept any regulatory body. The ICH guidelines and recent agency guidelines, the framework is calibrated against a Q series or quality and S series, stability series, plus the recent FDA and EMA AI discussions. Client should reference on one of these. So these all different guidelines should be considered. Recommended executive order. So here the order will be a month-by-month plan, TPP to target product profile to PPQ. 12 roughly 12 months, 12 plus months. Note the stability run in the background for 5 months and the digital twin comes online around 6 months. So active learning can start cutting experiments. First month will be TPP, QTP, CQS. First or second month will be API fee formulation. Second will be excipient screening. Third will be phase A DOE. Third, fourth is phase B DOE optimization, screening and optimization, and manufacturing optimization in process controls, dissolution permeability characterization, and 5 to 11 stability, 3 months, 6 months, 9 months, 12 months. 5 to 6 will be analytical method validation, 6 will be master ML pipeline, 7 to 9 active learning iterations. 9 to 12 is scale up engineering, and 12 plus PPQs. Okay. Next. The key difference every prediction task, the first choice algorithm and a backup. The rule of thumb at the bottom is one to remember. Always train a baseline along your champion to gain is measurable. So in task, first choice algorithm backup. Predict CQS, XGBoost will be the first choice. Second choice will be random forest. So predict stability, Arrhenius plus ML residual, pure XGBoost. Explain prediction, SHAP. Our second option will be a line. So these are the algorithms like ML models. ML models for particular model will be suitable. You should know which model is for the which data set. So the common mistakes will be with compound state, skipping stage one, skipping pre-formulation, optimizing before screening, pure ML with no mechanistic anchor. No uncertainty here. The poor data quality, the wrong cross validation. If your data is not that much good, poor quality is not there, your prediction also it won't give you good results. So data, pure data should be there. No data, the data is the fuel. Okay, so you should always pharmacy in pharma data is the problem. We won't get the quality of the data. That's why so many in experiments will be failed. So always focus on collecting the good data. Okay. So expected outcome will be 30 to 60% reduction in experiments, 3 to 6 months faster time to IND and IMPD. Higher will be PQ success rate, and better will be the regulatory doses. So this is the formulation. Okay. So this stage one is always closed to loop is never one shot. Mechanisms with first ML, second, and version data, version models. These are the 12 days, 12 stages. So everything should be included to get the good formulation results. Okay, and I want to show you one thing in silico semaglutide. So here in semaglutide, long back I did this experiment. So the in drug discovery, like this is the code. If I generate the code, every step, so target identification, target validation, hit identification, lead. So in different techniques, if you these parameters you should optimize it and you give the code here in graphical representation you will get these kind of results. These everything for everything will be coding is given. See here, phase one safety and pharmacokinetics monitoring. Phase one safety monitoring and phase one PK parameters, adverse events and vital signs, ECG, lab tests, Cmax, half-life and AUC. This is the frequency, pharmacokinetic, and this will be phase two efficacy and safety, efficacy endpoint, safety endpoints, and this is phase 3 enrollment and endpoint outcomes. And this is the different mild, moderate, severe, and serious adverse events. And this is the target identification, target validation, hit identification, lead optimization, candidate selection. So in each parameter, it will be evaluated LCMS, HPLC, ELISA, semaglutide development, method developments, and all. So this is like a usage and importance. PK/PD parameters for the semaglutide, and this will be preclinical development, focus area, pharmacodynamics, pharmacokinetics, toxicology, safety pharmacology, genotoxicity. So these like graphical representation, while see these are entirely if you studying it will take around 2 to 3 months if you take, but if you giving in a graphical representation, it will be easy for 2 to 3 hours. You will get it the entire results. These are drug discovery, pharma companies annual earnings. So Pfizer, Novartis, so based upon the data we are getting from the existing online data. So annual tablet consumption and common side effects reported. Okay. So these are this is a graph. Frequency, common adverse events, and these are the recent market share of semaglutide. This is the only peptide now demand is there. Projected pharma market growth and innovation impact. So this is the these are structured ML work if you did. If I want to see you one Excel sheet, okay. See, these Excel sheet, I made it. See, this is the this is the Excel application like this schema. This is called a schema. Everything you should be. This is actually 97 rows are there here. See, these are all Excel sheets are there. All Excel sheets. See, submission and products. If you see the product, see around 49,000, 50,000 cells are there. These cells. See here, the compounds. I said na CHEMBL compounds. These are the compounds up to 50,000. So these are the parameters. See the column 1, column 2, small molecules and the values. See here. So like this, you should generate you generate the data. You generate the data. See, check summary, transporters. These are the transporters. How many it is there? Around 560. So these are around 97 rows are there. These are ML applications. Like this, we should design it. Like this, you should prepare one Excel sheet. You should prepare one Excel sheet and you should run the model. You run the model and you should give the results in that way. And you should graphical representation to do it. Then only you can learn things easily. You can explain your students how the work actually works. If you do the projects, then only you can confidently can tell your students. Okay. Thank you. Thank you, ma'am. Any doubts? Please let me know. Hello. Anyone is there?
Sir.
Yes, thank you, ma'am. Please let me know any questions.
Okay, sir. Okay, sir. Audience, if you have any questions, please ask. Dear participants, if you have any questions, please post it in the chat group. Sir will answer the questions.
Good afternoon, sir.
Yes, ma'am. Good afternoon.
Actually, we are very much new to this techniques, sir. Actually, all faculty and everyone.
Yes, ma'am.
Like, can you give how to like learn all these things as a faculty so that we'll make the students to learn?
Yes, ma'am. So
Yes, sir.
actually, in whatever the content is given,
Yes.
the notes, the books given now, this pharmacy syllabus. Here
Okay.
the few books is given, ma'am. So for Python, for statistics. So for basic level and few websites also given. First you focus on that one. You can take this textbook, each chapter you take it out, you can go through it. If you are unable to get the things, you put it on ChatGPT and ask me how to get it in a simple manner. Can you understand me? Can you explain me in a proper, simple manner? It will teach you. It will teach you in a basic kids way. It will tells you. See, any topic, if we explain in a simple way, that is you are understandable.
Yes.
Any topic, any concept. If you are unable to explain in a harder level or any level, no use and all. If any harder topic, if you explain in simple manner in a kid's way, then that will be your understanding. So the books is just now is given and all. I download actually books and all. You first focus on these books, man. That is good. That is more than enough.
Okay, sir. Thank you.
Yes, thank you.
Thank you, sir, for your explanation. Anyone having question, please ask.
Very informative lecture, sir.
Thank you,
But
ma'am.
we will have to follow. Daily we will have to, practice.
Yes, ma'am.
Some
Every day,
other thing.
my interest actually, I will work
Very,
like
very
separately.
informative. So much of, data is given. Morning to evening. Thank you so much.
Thank you, ma'am.
Thank you, sir. Thank you for
Thank you.
sharing your knowledge, sir. So
Thank you, ma'am.
whatever the knowledge that you reflected in your presentation and the well-structured content of your slides were truly impressive, sir. Thank you for sharing your deep expertise and valuable industry perspectives. Sir, now we come to the end of the session. Now we have to honor the sir. Sir, we sincerely appreciate the time and effort you devoted to sharing your expertise with us, sir. Thanks you once again for your outstanding session and for making this FDP a memorable learning experience, sir.
Thank you for
Sir,
giving
we
me
will
this
honor you.
opportunity.
Sir,
Thank you, ma'am.
sir, due to
Yes,
the
yes.
sir, due to the power fluctuations, we can't display your certificate here, sir. I
Okay,
hope
ma'am.
you understand. We will forward the certificate to you, sir.
Sure, ma'am. Sure, ma'am. Thank you, ma'am.
Sir,
Can you provide
if you
the mail ID of the speaker? Prakash.
yes,
sir.
yes, ma'am. Yes, yes, ma'am. With permission from the sir, we will share the mail ID, ma'am.
Sure, ma'am.
Thank you so
Sir,
much.
can you please share the PPTs also, sir?
PPTs?
Yes,
Sure, ma'am.
PPT.
Sure.
Yeah.
If possible, sir, if possible, you
Sure,
can
ma'am.
share.
Actually, what kind of information you want, or what are the topics if you want, now I will prepare it and I will give it to you, ma'am. Suppose if you anyone
Okay,
required
sir.
anything will be there. I will assist you.
Yeah.
Okay, sir.
Definitely.
Okay.
This is actually a generalized whole thing, ma'am. Can't for actually for 5 years or 4 years students, na, this will be generalized way. If you ask
Okay.
to go through it every topic
Specifically.
deeply, yeah, specifically.
Sir, even it is generalized PPT, it is well framed and
Yeah.
it is giving so much information, sir.
Thank you, ma'am.
Like ABCD, we got more information starting from the drug discovery to the marketing. What
Marketing.
type of MLs we are using. We got basic idea from your presentation. Thank you, sir.
Thank you very much.
Okay, thank you, sir. We are ending the
Thank you.
session. Thank you, participants. Tomorrow we
Sure,
will
sure.
meet in another session.
Thank you, ma'am.
Thank
Thank
you,
you
sir.
very much for giving me this
Thank you,
opportunity.
sir.
Thank you, ma'am.
Thank you, sir.